All right, good morning, everybody. I think we've got a full house. This is great. Next year, we're going to need to add more seats, I think, which is a good problem to have. Hi, everybody. My name is Stephen Bell. I'm Emily's husband. I'm Madeline's father. Together with Alice Fox, her son James, and her husband Duke, we formed the Bow Foundation 5 years ago. And at the time, it was really a pipe dream to be able to pull together a group like this. So it's great to have you here with us today. Thanks for being a part of this. Really, this doesn't happen if each of you doesn't make the commitment to be here with us. And that makes today so much more special. Welcome to St. Louis. We're glad to be here. And I want to take a moment to thank all of the parent volunteers who made this possible. We had a steering committee of parents who helped Emily and Alice coordinate this conference. So I know it's embarrassing, but if I say your name, I'd like you to stand up and be recognized. So Betsy McNamara, Dan Habib, Elizabeth Hausman, Lacey Ann Bauer, Christina Walker, Peter Slutsky, Shelley Van Dersen, and Stephanie McMorris. So thanks to each of you for your support. Like I said, not possible if you're not here helping us make this happen. It's also not possible if WashU and Dr. Gurnett don't help us and make the space available and work with us to facilitate the clinics, right? It takes two to tango, and they're a huge part of this process. So thank you for that. Thank you, Dr. Garris, for traveling and being a part of the clinic. We appreciate you being here. Thank you, Dr. Axine, for your work on behalf of GNAO1 families and for tolerating what I know were travel complications and challenges. A few housekeeping items today, just as we get rolling. There are tables available in the vestibule of the auditorium and in each of the restrooms to facilitate easier changes for each of our young ones if there's a need. The anteroom directly behind this barrier is a place where children can go and be a little more free if they want to. This session's being streamed in there, so feel free to make use of that if you have a need. I know we've got a lot of young ones here, which is great. We're gonna have a morning break a little later this morning, and I hope we can all gather in the main lobby to take a picture. It's a great, powerful tool kind of to showcase what we're able to accomplish together. Stephanie McMorris is taking pictures, and she's also collecting pictures. So please take a lot of pictures. We don't have a professional photographer today, but we've all got phones, and I think we'll make the most use out of it. We use those pictures a lot on our webpage. We make sure that we feature them strongly on social media. So we appreciate you, Stephanie, for helping us with that. And then just two quick closing notes. This isn't the end of the fun today. We'll continue the clinic on Monday. And then we also on Monday have a virtual number of research presentations with researchers from the Netherlands, from Israel, from Italy, from the USA and Switzerland. And if you haven't registered for the Zoom link, Emily's happy to get you set up with that. And then on June 7th, we'll have a virtual roundtable moderated by Dr. Axine and Dr. Newbig to focus on fostering collaboration in the medical community about alignment and GNAO1 research, just to make sure we're all talking and the researchers are coordinating and collaborating so that our funds go as far as they possibly can. So with that, Emily and Alice, thanks for being here. Appreciate all you're doing. So we'll get rolling today with Dr. Axine, and I have to start by noting that Dr. Axine had a couple of flights canceled and a couple of flights tremendously delayed. So she's joining us virtually today from Charlottesville, Virginia. Not what I think she wanted, but we appreciate her flexibility and her willingness to still participate. Dr. Axine is a neurologist at the University of Virginia in Charlottesville, and she specializes in evaluating and managing seizures and epilepsy in children. She was born and raised in Ames, Iowa, where her father was a college professor. Axine studied biological sciences with a minor in art history during her undergraduate, and she completed her medical school at the University of Iowa before moving to Charlottesville for residency training in pediatric neurology. In her free time, Axine enjoys hiking in the mountains with her family, and Dr. Axine will turn to you for your presentation.
There we go. All right, can you hear me now?
Yes.
Okay, good. Well, hello everyone, and thank you for that introduction. As Stephen said, I tried very hard to be there for the clinic and for this presentation, but the travel gods did not agree, and I spent an unpleasant night in Charlotte. But I'm happy to be here today, virtually, talking to you a bit about what I've been working on and my next project. So as Stephen said, I'm a child neurologist, but I focus mostly on epilepsy. So I'm interested, of course, in all children with GNAO1, but really one of my big goals is trying to learn more about epilepsy within GNAO1. So firstly, it's just a note on what we call this. I'm sure you all have seen lots of different labels. GNAO1 related developmental disorders is what I've used in my publications, GNAO1 NDD, but you'll also see GNAO1 encephalopathy, other related disorders, GNAO1 related dystonia. All of those are the same thing. It's all just a label, right? And so I don't have a preference, but just so you understand that there's different words you'll hear. So I know Dr. Newbig is going to speak much more eloquently than I about the basic science of GNAO1. I summarize it in saying it's complicated, right? So GNAO1 is the name for the gene, which if there's an error in how the gene is spelled, it creates a problem in the protein. So that's what's pictured here in this pretty colored model that was done by some colleagues up in Boston. When there's an error in the gene, it causes an error in how the protein works. And that simply, I think the simple way to think about it is that causes problems in the way GNAO1 can communicate within a cell. And so Dr. Newbig and many others are trying to figure out more details about that, but it's complicated. And so basically it just changes the way it works in the brain, and that can create the features that we see in GNAO1. So my focus is really on what we actually see in children and adults with GNAO1. So it's heterogeneous. Heterogeneous is a fancy word for saying it really is widely variable, as you all can probably see when you're together. So epilepsy or seizures was what was first described in the first publication back in 2013. So epilepsy is a tendency towards seizures. What you may see, we'll talk about a little more, is that many children with GNAO1 that have epilepsy have a developmental and epileptic encephalopathy. So 52% of children with GNAO1 have epilepsy. Then a movement disorder, what was described after epilepsy was first described in GNAO1. And generally what's talked about in the literature is those cases that are quite severe and progressive and cause a significant amount of disability. But 76% of people with GNAO1 will have a movement disorder. And then there's this sort of middle ground where you may have some seizures and some movement disorder with varied features of both. So the caregiver survey that was published last year was an effort with me, and I got to shout out Emily Bell, who did a lot of work with me to get that published, was trying to ask all of you who know what your children look like to contribute to us to describe what do these children look like. And so we were able to get 82 people respond to the survey, which is a huge amount considering that it is an ultra rare disorder. You can see here on the slide that it was an international effort. So that was really exciting to have people from all over the world contributing information about their children. And it's interesting now, kind of, several years later from when we started gathering this data, 82 is a great number, but I know from a colleague in Germany that they have, well over 20, even just within Germany. So there are more of the children out there that are not described in this study. But just briefly, the questions that we asked in the survey were things like, what is the first thing that you were worried about in your child? What was the first abnormal sign? And here you can see what you all see is that for many children, it was things like hypotonia or low muscle tone, floppiness, developmental delay, and 67%. And that's often related to the floppiness. But the cardinal features that we often focus on, things like seizures and abnormal movements, that was much less frequent. So seizures, 29%, that was the first sign of something abnormal. Abnormal movements, only 20%. So another question was, what age were you first concerned about your child? And it's clear that it presents early. There are early signs that there's something different and unusual about the child. So really 71% before the age of 6 months, and pretty universally, every parent is worried about their child before age 1 if GNAO1 is the cause. Diving in a little bit to looking at the movement disorder and the epilepsy, you can see epilepsy is featured in purple here, which is usually the epilepsy color, and movement disorder in green. This is looking at age, and so. And on the y-axis over here, you see the number of participants that agreed to this. So what we see here, first looking at the epilepsy, is that you see there's more participants that marked that the epilepsy started below age 1. Okay, you do see some purple above age 1, but those numbers are lower. Within the movement disorder, you can see that it's a little more evenly spaced. There's quite a few that presented before age 1 with their first abnormal movements, some more between age 1 and 3, less so after age 3. So diving in a little deeper to below age 1, again, with the purple with epilepsy, you see that most of those kids, 12 of them, presented before the age of 3 months, with less later within the first year. Whereas the movement disorder, they all look kind of the same, meaning, so what I take from this is that seizures are more likely than movement disorders to be the first presenting sign of GNAO1, but movement disorders also present young, usually below age 3. So our major findings in the paper that we had is that GNAO1 is often a non-specific clinical presentation. So what that means to me as a clinician is that if I meet a child in clinic, I'm not able to look at them and say, aha, you have GNAO1, right? It could be a symptom, the symptoms could be consistent with multiple other disorders. But that universally parents do notice something unusual about their child before age 1. Movement disorders are generally more common, but this is often not the first sign that there's something different going on. Epilepsy can be severe and presents early, but these cases, and I'll talk about this a bit more, are often milder than what is described in the literature. There were some limitations to my study. It was really caregiver report only, and although you guys are the expert in your children, it's really hard to figure out exactly what kind of seizures your child has, and it's complicated within GNAO1 in general. And we didn't have any medical records, so I wasn't able to actually go and look and see what the clinicians that are taking care of your children actually thought as well. So I took that information to heart in designing the next study. So switching gears a little bit to epilepsy and GNAO1, as I told you, it's less common than the movement disorders, and there's less publications about it. So there's less for us to build on because of that. Up in the top right, you see, this is sort of explaining what a developmental and epileptic encephalopathy is. That's a big word, so DEE, right? So what does that mean? So GNAO1 is the cause of this, right? And so GNAO1 can cause epilepsy, and it can also cause developmental impairments. But those are all related. And so we focus a lot on treating the epilepsy in hopes that the development will improve, or will that a child will reach their peak maximal development. But even if you fix the epilepsy, let's say we are able to find the perfect combination of medications, treatments, the developmental impairment remains and probably would have been there all along, right? So the point here is that it's all interrelated. And I think this is relevant for those that have primarily movement disorders as well. But within the epilepsy community, a DEE is how we kind of describe this concept. So lots of seizures, difficult to control seizures with developmental impairment is generally what that means. So in the caregiver survey, you all responded that about 52% had epilepsy or seizures. So epilepsy is the tendency towards multiple seizures, or I also was interested in whether people had any seizures at all. So 52%, 27% reported that they struggle with ongoing seizures, but 25% actually said there's no seizures happening right now. Okay, so that was interesting to me to hear that, you know, although we really do focus a lot, as we should, on the very severe epilepsies, there's a proportion of people that don't have continually medically refractory lifelong epilepsy. And I think that's important for parents to know too, is that we don't really know what the future looks like in children that have a lot of seizures. 3 of the participants actually said they had never been on an anti-seizure medication, which really speaks to them not having very many seizures. So looking in what's described in the literature, the first report was in 2013. So looking at everything that's been described from then until now, there were 22 different papers describing GNAO1 neurodevelopmental disorder with information that I could extrapolate. 88 individual children or adults described in there. 38 variants, meaning 38 different locations in the gene that cause their disorder. 85% of those were described to have a movement disorder, 54% with seizures, and 37% with an overlapping disorder. So those numbers are pretty similar to what we got in the caregiver survey. So that's good to know that those numbers align. But diving into the seizures, of those with any seizures, the majority could be categorized as the DEE. There were some that described focal seizures and generalized tonic-clonic seizures, but 31% of all children or adults with GNAO1 were described to fit into that category of the DEE. So I know there's a lot on this slide, and I could give a whole separate lecture that would probably bore you all to tears about seizure classifications and epilepsy classifications. But basically, what I want you guys to understand is that you can have basically any type of seizure with GNAO1. They can be seizures that start from one part of the brain, or seizures that involve the whole brain. So focal, one part of the brain, versus generalized, the whole brain. And all of those are described in publications. So when we decide what seizure type it is, then we just decide what epilepsy type, and that feeds into the epilepsy syndrome. So the epilepsy syndrome is the DEE, and you see listed there some words that some of you may have heard, things like infantile spasms, West syndrome, Lennox-Gastaut syndrome. Those are all subtypes of DEEs and can be categorized into that. When looking at all of this literature, my take home for you all is that there's no single seizure type, no single epilepsy type or syndrome with GNAO1. And so that makes it somewhat challenging to understand how, is there a single best treatment for GNAO1 because it's so varied. Generally, we do see that if you're going to have epilepsy, it's more common to be the early onset developmental and epileptic encephalopathy. So just switching gears a little bit to how do we treat epilepsy? Unfortunately, the holy grail is that there's a disease modifying treatment, a cure, and of course that does not exist at this time. So our focus primarily is on medications. Largely, it's anecdotal experience, meaning whichever doctor you're working with presumably has training in how to use these various medications, and you all decide what is the best medication for my child. There's lots of publications, case reports, oh, this medication worked better than this one, but in the literature, there is no best described medication. In my interactions with probably some of you in that room, asking the question of what is your experience with medication, during our virtual 2020 clinic, every single parent or caregiver picked a different medication that was the most effective. So those included CBD or Epidiolex, lacosamide, Vimpat, Topamax, topiramate, clonazepam, which is also used for children with abnormal tone, and dietary therapy. So of 5 people I talked to, everyone picked a different medication. There's a whole longer list here of medicines that have been effective, although certainly none of those were ones that made the children seizure free. And then of those that were rated as not effective, you can see some overlap. Some people thought CBD was completely ineffective, whereas others said, oh, that was the best medication for my child. So that's really frustrating, right? It's frustrating for everyone. It's for me taking care of these children, and also for you all saying there's no single best medication from that type of survey. When I surveyed in the 82 participants from the GNAO1 caregiver survey, pretty similar in that there was no single best medication that was the most effective. And I also saw that of those that had been on a medication, it was on average that they had tried 3 medications with a range of 1 to 8 medicines. So we need more information about this. When medicines don't work, then we call it medically refractory or pharmacoresistant. We start thinking about what else can we use. And these are also options when we're really frustrated by the side effects of medications. So thinking about the ketogenic diet is important. I'm very interested in looking into this more. There's some published reports. There was a child that reportedly became seizure-free. And those that I've spoken to as part of the natural history study, it was pretty mixed in people's experience. In the caregiver survey, very mixed in terms of people's experience. Anecdotally, I've worked with a family who felt that the diet actually made things worse. And we really also don't know if there's a potential benefit of those that have primarily a movement disorder focus. So this is something I'm interested in looking more into, trying to understand this. A vagal nerve stimulator is another non-medication type of treatment. It's a surgery where they implant a small computer into the patient's chest that communicates with the vagus nerve, which is in the neck, that communicates with the brain, and can reduce seizures. Deep brain stimulator, you guys probably all have been familiar with as a treatment of abnormal movements, and certainly the results for that, and some of you all has been profound. There's also data for that in helping seizures, which I know my colleague Dr. Goodkin spoke on a few years ago at the last in-person conference. And certainly there's evidence that that's helpful. So it's interesting, of those that have had a DBS implanted for their movement disorders, did it help their seizures? And from some of you, you've told me that actually maybe it did. So that will be interesting as we get more information about that, to see if that can be helpful for other patients. So in summary, of GNAO1 epilepsy, within GNAO1, about half will have a seizure or epilepsy. Not all epilepsy is medically refractory, resistant to medications, or lifelong. Epilepsy does often present earlier in life, whereas the movement disorder may develop more over time. Early onset developmental and epileptic encephalopathy, the DEE, is the most common type of epilepsy syndrome. And treatment of GNAO1 related epilepsy really should be targeted towards the type of epilepsy, rather than kind of a general, like everyone with GNAO1 responds to this medication. That's not the information we have right now. So it really should be in discussion with your doctor about, well, what type of medicine works for this type of epilepsy. And really, we need more data. This study, or this slide, is borrowed from some ideas from Dr. Viehover, looking at how do we actually study rare disease, right? So the first thing that happens is a case report describing the disorder. Then we get case series, more groups of patients with the same thing. And then what you really need is natural history studies. So this is to define, what does GNAO1 look like? Because you can't come up with something to treat a disease modifying treatment if you don't know what kind of the baseline presentation for a disorder is, right? So that's why all of you that are there in the research clinic are there. That's a prospective natural history study looking forward year to year. How does this disorder change over time? And then the other piece of that is a retrospective natural history study. So looking at, your child is 5, what has their life looked like so far? And grouping lots and lots of people together, we can learn about what is the natural history of the disorder. So we really need both of those pieces, the retrospective and the prospective. And I think in general, the epilepsy aspect of what we have right now is very lacking. So that's an interest of mine. So this is just a brief description of what I'm doing in my new study that's just launched. I am looking to enroll anybody with a diagnosis of GNAO1 in a research database that I'm developing. What I'm going to do is try to identify the phenotype. That's a fancy word of saying, what is the clinical presentation? Using both a questionnaire built on what I did before, but a little more advanced, easier, more user-friendly, hopefully, and comparing that with medical records, validating it with what did the MRI show, what did the EEG show, what did the genetic report say, with the goal of coming up with a genotype-phenotype correlation. So putting together where in the gene is it, and what is the clinical presentation. So I was hoping to hand out a stack of these flyers in person today. The flyers are in Virginia, not in St. Louis. So I'm happy to send this to anyone, and we'll probably try to email this out. But I am recruiting for this study. So anyone with GNAO1, I would love to get you guys enrolled in the study. It's going to be a natural history study of GNAO1, all virtual. It'll be first a phone call with the study coordinator to do consent and talk about all the details of the study. You'll be emailed a link to fill out the questionnaire. And then there's a spot on the questionnaire where you can submit medical records for our review. I know many of you probably already have those medical records in a PDF, and that's easy to just submit them. If you don't have them, we can hunt them down for you. And then I'll be going through all the data and putting that together. My hope is to get everyone that's involved in the natural history study, the research clinic, because we are actually able to link your data from there with the data that I'll be gathering. So Dr. Vhover and her team are able to see that as well. So this is a really important addition to that study. Also, the exciting part is for those that aren't able to be in the in-person clinic, we can include you as well. So anyone with GNAO1, we want as many people as possible. Right now, it's English language only, unfortunately. But if there is a language need, just reach out to us and see if we can accommodate that. What my goal is that we will be able to provide a de-identified, meaning that nobody knows who you are, but we have a big database of GNAO1 phenotype and genotype correlations that we can actually share with researchers. I'm a clinician. I don't do the benchtop stuff, meaning like I'm not in the lab trying to figure out how GNAO1 works. But we've already had some people reach out with interest in the data that we'll be getting so that benchtop researchers can understand how this actually looks clinically. So that's what I'm hoping to help with, as well as just describing, a broader spectrum of what the disorder looks like. So my study coordinator's name is here. Her email is there. I know it's a long email, so that's something that myself, Emily, the team at the Bow Foundation, we can all get to you. There's a phone number as well. I know my study coordinator's out this week, but if you call or email, she'll get back with you when she's back. So hopefully all of you will participate. So I first want to say thank you to all of the study participants from past study, future participants, anyone with GNAO1, the efforts that you guys have done to move the research forward is why you guys are all there, why this is all happening. So you guys should be commended on that. Thank you to the Bow Foundation for funding for the research project, but also just you guys are working so hard. For those of you that don't know the Bow Foundation leaders well, you guys should be really proud of how organized and the efforts that they put together are really phenomenal. The collaborators for the caregiver survey, the Bow Foundation, as I said, Emily Bell really helped a lot to move this forward. Thank you also to my mentors. Many of you might have had the opportunity to meet Dr. Goodkin at the last clinic as well. And you can see here, it's my husband and kids kayaking. Instead of being there doing the study, that's what I did last night, is went kayaking because I was so upset about not being there. But hopefully we'll all be able to meet in the future.
Thank you so much, Dr. Axine. Really appreciate your continued focus and partnership on GNAO1. Doesn't happen in a vacuum. And I know Emily really appreciates your continued partnership with her and with us on this GNAO1 process. Thanks so much.
Absolutely.
I forgot to mention in my housekeeping note that Mary Hecht, she's a child life specialist here at St. Louis Children's. She'll speak with us this afternoon, but she's currently in the ante room behind those partitions, and she's got some kids craft sessions that she'll be organizing through the morning break. So if you have an interest and you have a desire, there's something for y'all to do in that room beyond just letting your kids walk around. Yes, Emily? That's a good question. Does anyone have any questions for Erica before we move on to the next speaker? Happy to bring a mic out if so. Okay. Thanks so much, Erica. Look forward to being a part of that research study as that launches. Todd, if you want to come up and shift, feel free. Our next presenter is Dr. Richard Neubig, MD, PhD at Michigan State University. He's a professor of pharmacology and toxicology. Dr. Neubig has been a longtime friend of the Bow Foundation and GNAO1 research. He's an accomplished expert in the field of G protein coupled receptor signaling and intercellular modeling of that signaling. G protein coupled receptors are the targets of about 30% of the current drugs, and Dr. Neubig is helping discover new ways to control their function. The Neubig Lab is an interdisciplinary lab and is spearheading a wide range of projects really focused on understanding G protein coupled receptors and how they interact and how the implications are for our children. So with that, we'll let Todd get the presentation pulled up, and then we'll invite Dr. Neubig up to deliver his remarks. Thanks so much for being here.
Let's see. Right, pointer. Yeah, yeah, laser. is good. There we go. Perfect. Okay, well, thanks. I'm delighted to be back here again. Delighted to be back here again. The Bow Foundation has really kept us in the game with GNAO1. We started on it quite a while ago and struggled to get NIH funding, but the Bow Foundation has kept us really working on this, and I'm excited to be able to share some new data. It just came on Monday, and my team pulled it together, and I'll be able to show that on gene therapy approach, which I think is potentially very exciting. I'm going to end up getting a little technical here. It's important. I'll try to do it at a level that will be understandable, but I'd be happy to answer questions. I think what it relates to mostly is what Dr. Axine talked about in terms of the complexity and heterogeneity of GNAO1 disorders. That there are groups of patients who have the same mutation, the same allele as we call it, and they may respond to the same therapies. Others with a GNAO1 disorder, but a different mutation or a different allele, may respond to completely different treatments. And I'll be touching on that in the talk, and how some of the different categories of function that occurs with the mutations may require specific different treatments. So I think that'll be very important going forward. And I think Dr. Axine's genotype-phenotype correlation, as those numbers expand, will help us a lot in the therapeutics development, in terms of how to come up with the right therapies. So I want to mention that this is a close collaboration with Dr. Miguel Estevez at UMass. He's an amazing gene therapy researcher who has actually gotten gene therapy in the clinic for Tay-Sachs. And so he really understands what it takes to move from my lab, which is a bench and mice, into people. So I think that's a very valuable collaboration that the Bow Foundation has really allowed us to kick off. So as you know, the GNAO1 disorders include epilepsy and movement disorder, as well described by Dr. Axine. There also is an overlap. So in 2013, the Nakamura et al. Described the DEE-17, this very severe epilepsy, which is present in some children. Others have more mild epilepsies. The onset was very early, as Dr. Axine talked about. Later on, it was discovered that the movement disorder was also present and actually more common in the GNAO1 group. And it includes a number of things which Dr. Axine talked about. Later onset, typically milder course, but also quite detrimental. But there's specific mutations or specific alleles, as we call them, that were found in patients with movement disorder who did not have much in the way of epilepsy. Mutations in R209 and another one here. So here's one of the first papers on this. There's a series of several papers that came out describing the movement disorder. And of the first 6 patients that this group described, 2 had mutations in R209 and 4 in the E246K. What I'll be talking about is a mouse model that we generated with Bow Foundation support that carries this R209H mutation. We've tried to make mouse models with about 6 different mutations, and so far, almost all of them have died. We've just not been able to create the mice. But the R209H, we were successful, so we're now able to go ahead and do studies with that mouse model. So as I said, we're going to dig a little bit into the signaling mechanisms and how mutations can change the function of a gene. So we're working with the G proteins. The GNAO1 is one of 20 different G proteins. This connects signals from neurotransmitters and hormones through a receptor, through the G protein, into what we call effector molecules. So that the effector sends the signal inside the cell to cause the neuron to fire faster or slower, or grow faster or slower, extend processes or not. So we have a signal that starts with a neurotransmitter outside the cell to a signal inside the cell. Now, when you have a mutation in a gene, especially one of these G protein genes, it can do a lot of different things. Typically, mutations cause loss of function. So in most disorders, like storage diseases like Tay-Sachs, the mutation just kills the activity of the protein. It's called loss of function, or LOF. So it just shuts down the function of the protein. And there are a number of mutations in GNAO1 that do this. They cause loss of function. Those tend to be more associated with epilepsy. There are also a possibility with the G proteins, especially, where when you make a mutation, instead of shutting down the function, you make it have too much function. And so obviously, if you have one mutation that causes the gene to not work, another causes to work better, you're going to need different treatments for those two different things. And then there's another category that has been implicated in GNAO1 more recently by the Martemyanov lab in Florida, and that's called a dominant negative. So really, this is a loss of function where the G protein doesn't work, but not only does it not work, it also blocks the function of the normal gene. So almost all patients with GNAO1 have one normal copy and one abnormal copy. And so why doesn't the normal copy keep things going well? Well, if you've got a dominant negative mutation that loses its function, but at the same time blocks the function of the normal copy, then that can contribute. And the different treatments, both at the level of pharmacology and at the level of gene therapy, are different for loss of function or gain of function dominant negative. So I'm going to be getting into that. And I apologize that this is complex, but as Dr. Axine said, GNAO1 is complex. It's not like a storage disorder where you don't have the enzyme, you put the enzyme back and everything goes well. If you have too much GNAO1, it may do one thing, and if you have too little, it may do the other. And we have to strike the balance of getting that just right as we consider therapies. So why is this important? Basically said this. Gene therapy treatments are probably going to be different when they're different molecular mechanisms of this sort. For loss of function, probably like in Tay-Sachs, you just replace the gene and it will hopefully work. For gain of function and dominant negative, you may need to both replace the gene and eliminate the bad copy, eliminate the copy that's overactive or is blocking the normal function. So these are considerations that we need to think about as we do this. One of the complexities, though, is we don't completely understand for a particular allele whether it's purely loss of function, purely gain of function, or dominant negative. So that's a controversy in the field that we're discussing among the researchers to really better understand that. But in gene therapy, the good news is that replacement can be helpful for a significant subset, and the combination hopefully would be able to be valuable in the other subset. So to look at this, we have two animal models. I'll be mainly talking about the R209H mutant mouse, which was developed with Bow Foundation funding. We went in with molecular scissors, CRISPR, and changed one copy of the GNAO1 gene to have this R209H mutation. This is a somewhat mild mutation, but it can cause fairly substantial movement disorder. The mice do very well. They're completely viable. They breed well. What we found, though, is that they have hyperactive movements. They run around more than normal. They're hyperactive. Now, that may or may not relate exactly to how a human being would do that, but it gives us a functional readout that we can use when we try a therapy of some sort. Can we suppress that hyperactivity? And that will be telling us that we're able to modify the function of the gene in a way that should be beneficial. We also have another mutation that's a loss of function where it has a mutation in a splice site. It's not like this isn't present in patients, but it does cause some adult lethality, some seizures, but a seizure and movement disorder. And I'm just going to show you a little bit about what we see in the mice. So this is the hyperactive R209H mutant. And when we put them into an open field, so this is just a box, you put the mouse in the box, and they run around, and you have a little camera that follows where they go, and this shows how it runs around. For a mouse with a normal GNAO1 complement, they run around like this. The R209H, they run around a lot more and for a longer period of time. Typically, when you put a mouse into a new environment, a novel environment, they run around a lot at the beginning and then not so much later. But the R209H mice keep running and running. We've also been able to test seizures. We used an old method that took us 3 weeks to assess the seizures. That was very challenging, but with the Bow Foundation support, we were able to get a thermal seizure apparatus. And what we found was that the R209H mice did show some seizures. The way this works is we basically give them a fever, a high fever. So we raise their temperature using a heat lamp and a thermal controller to up to 42.5 degrees Celsius, which is quite high. And then we hold it for a while and see if they have a seizure. So the mutant mice had some seizures, but even one of the wild type mice, normal mice, had a seizure. When we looked at our other mouse model that has the loss of function, almost all of those had seizures and at earlier time points. So we do see a difference between the movement disorder mutation and the epilepsy type mutation, the loss of function mutation. These mice also have a movement disorder. They don't have the hyperactivity, but their walking is not normal. Their gait is abnormal. When we put them on a treadmill and look at how long their steps are, they can't maintain the normal step level. So they do have some movement abnormalities. And this mouse is being studied in a patient, in a parallel study of gene therapy related to, funded by the Children's Genetic Research Group. But I'll be talking today about this one. So one other key factor, and I think this is a really important research question that we really need to nail down, is what part of the brain is controlling the behaviors we're looking at. Because if you're going to do gene therapy, you typically don't treat the whole brain, you treat a region of the brain. So understanding which part of the brain is controlling the movement disorders or contributing to the epilepsy will be very important, and we just don't fully have the answer to that question. We know from other studies that the hippocampus and the cortex, which are large areas on the outside of the brain, are critical in epilepsy. Some focal seizures have hippocampal neuron loss. There's a lot of research on this. Also in movements, the striatum, which is a more interior part of the brain, is very important in movement. It's the key site of abnormalities in Parkinson's. You have neuron loss in the striatum, and you end up with the opposite of hyperactivity. You end up with slow movements or lack of movements. And in some hyperkinetic movement disorders, the striatum has been implicated also. And this is where we're focusing our work, because the data from the R209H mouse appears to potentially be from the striatum. It's also a site of DBS. So for deep brain stimulation, one of the key targets for deep brain stimulation is the GPI, or globus pallidus interna, which is a part of the striatum. So there's a lot of reasons to think that the striatum may play a role in that. And that's where we focused our gene therapy studies. So for the gene therapy, what are we going to do? So we've been working, so Dr. Estevez's lab has created several different types of gene transfer. So the way this kind of work is done is you introduce the gene you want to provide to the patient into what we call a vector, which is essentially a virus that's been modified so that it can't really make an infection, but it will locally get into the neurons and will provide the gene to the neuron and have the neuron make that protein. So the first thing we did was using a tagged protein. This allows us to figure out where it is. But unfortunately, the tag messes with function. So we can't use that for a treatment study. For the treatment study, we have the untagged versions. And we also have two genes. We have GOA and GOB. Everyone talks about GNAO 1.1, but it turns out the GNAO1 gene can actually make two proteins from the same gene. And figuring out which of those may be more valuable as a gene therapy is one of our goals. So far, we only have data on the predominant one, GOA. as I mentioned, putting in more of the gene or more of the protein will help the people, the loss of function situation, but it may make the gain of function situation worse. So we also want to be able to, in certain situations, suppress the hyperactivity. And to do that, the Estevez lab created what we call a microRNA, which makes an RNA that interferes with the gene expression. So we can raise it up with the gene replacement, or we can knock it down with the microRNA. And we can potentially do both at the same time, knock down the abnormal gene and replace it with a normal one. So those are all possibilities. Again, it's complex. Today, I'm just talking about the simple gene replacement with the GOA, because we have some pretty exciting, very new data on that. So I think I've already mentioned this. We want to increase expression of the normal protein, knock down the endogenous, or do both. And this should work for the loss of function. It may be valuable in the dominant negative. If the dominant negative is blocking the normal gene, if you put in more of the normal, it may overcome that block. So the first thing, the one that we've got data on already, should work in loss of function, may be beneficial in dominant negative, but for gain of function, we need the knockdown. So we're working on that. So is this even feasible? You know, we're talking about changing genes. Well, you know, a lot of you are aware of the deep brain stimulation. You can go in and put electrodes in. Well, you can do the same thing with a viral vector like this, the AAV vector, and introduce a gene therapy system into a particular region. And people have already started doing this in Parkinson's in the striatum. So this is a part of the brain, the striatum. The cortex, we think, is involved in epilepsy and other places, but the striatum is one place we think may be contributing to the hyperactive movements. And people are already doing gene therapies there. So it is absolutely feasible. And the vectors that the Estevez lab has made are humanized vectors. They are very much along the lines of what he would see moving into the clinic. So he's done this, as I said, he's done this for Tay-Sachs. And so his design is one that will work in the mice for our studies, but also could be translated to the clinic if we could get enough data to support the safety and utility there. So first thing we wanted to show is, can we get this into the striatum? So this is a control of scientists. We always have to have controls. We have to make sure we know what we're looking at. So if we don't put in the gene vector, we see no expression of the tagged protein. But if we put in the vector, we get a fairly nice expression. This is sort of this is the striatum here. We get a little spillover into cortex, but that's not all a bad thing. But we're definitely getting expression in the striatum. And the way we did that was working with a Parkinson's group at MSU, who do gene therapy treatments in mice. They injected it straight into the striatum, and we got expression. This is a map from the Allen Brain Atlas. This is the caudate putamen. You can see that's the region where the most of the protein is expressed. So this is sort of the punchline, and I saw this Wednesday afternoon. I was thrilled. So what we have is the movement in that open field. So we have mice with a normal GNAO1. They have moderate activity. They run around this distance. If we have the mice carrying the R209H mutation, we have two controls. One, we just injected saline instead of the vector. The other, we expressed the AAV vector carrying an irrelevant protein, a GFP protein. And both of these controls showed exactly what we expected, hyperactivity in this open field test. But when we put in the GOA vector, we saw a pretty clear reduction in the movement of the mice in this model. It's not completely corrected back to zero. We don't have enough. There's a preliminary study, the first round. We don't have enough data to know if this is statistically significant, but it looks quite clear to me with this. And so I was thrilled that with our very first try, the very first dose we picked, that we actually did see a difference. Whenever you do something like this, there's always an optimization process that you go through. And so we're seeing positive results. So what are the next steps? We've got a number of other behavioral readouts of movements, balance beam, hind limb clasping, walking on a rotating rod, walking on that treadmill, and then also thermal seizures. So we're collecting those data now. We'll have those compiled probably within a couple weeks. So we'll see that. The seizures is really important because one thing we definitely don't want to do is we don't want to introduce seizures in a mouse that doesn't have seizures already. So this is really a safety question, and we'll be getting those data as well. We're also going to be doing additional mice. We'll use the GOB vector as well as GOA. Does it do better or not so well? Then we're going to be doing the knockdown and knockdown replacement. So this has been a great project and really only possible because of the support of the Bow Foundation. So Jeff and Alex in my lab have done virtually all of the work on this. Jade and Cassie had done the earlier studies on the R209H. The Estevez lab at UMass has created all of these vectors and is studying them. The Sortwell lab in translational neuroscience at MSU did the injections for us. And of course, the funding from the Bow Foundation made this all possible. I'm delighted to be here. I'm really excited. Those little red bars just made my day, I'll tell you that. It's just the first step, but it's going the right direction, which is really thrilling. Be happy to take questions. Yeah, so the question was, if you have a patient who presents with both movement disorder and epilepsy, how do you know if it's gain of function or loss of function? It's right now we're trying to sort that out. And it's very, very challenging. So I think that I think knowing what the mutation is can give us some clues as we do more studies in the bench, either biochemical studies or mouse studies or zebrafish studies or other model studies that can give us some clues about that. It's still a controversy. So my lab came up with the gain of function model. The Martemyanov lab is really pushing the dominant negative model. And I think, there's controversy about that even at the level of where we are. But I would say if, it's going to require more work and it will be dependent on the very specific mutation and Dr. Axine's genotype phenotype correlation data. The more data like that she has, the better we'll be able to think about how to approach those things. Okay, thank you. Oh yeah, another question, sorry, yes. Yeah. Yeah, yes, I will. So the question was, can you measure loss of function and gain of function in, say, a mouse model? It's actually fairly complicated. GNAO1, unfortunately, is one of the most complicated systems that one could imagine. We have, and part of that depends on which neuron you're looking at. So a loss of function in an inhibitory neuron will cause stimulation. A loss of function in an excitatory neuron may cause inhibition. So it is complicated. The Martemyanov lab has some data in striatum with respect to cyclic AMP regulation. And so there are ways, but it is not trivial to do that. One more, yes. Yep. Yeah, it's a good question. So the Katnev lab in Switzerland has created another mouse model that appears to be viable. We tried the G203R mutant, And that one we thought we had, but then it turned out we had the G203R mutation, but there was also this splice mutant that basically killed function. So the only mice that made it through were ones where we disrupted the effective mutation we wanted. And Ketnev lab also found that the G203R mice were not viable in their hands. I think there's another approach which is challenging, but I know is being done, is to make a conditional mutation where you introduce it, but then you can turn it on by treating with a chemical that activates the mutation. It's challenging. There is one group that I know is working on that, and that may be a way to bring in more of the mutations that are possible. Yeah. Yep. Yeah, actually, the question was, what are the next steps? So, this is a very early data. Dr. Estevez really could probably answer that better. He's the one who's really been involved in the gene therapy clinical trials. Unfortunately, number one, it's a lot of money, which I'm sure you're aware of. But I think that the key things are that you need to demonstrate efficacy and you need to demonstrate safety. So we will get some safety readout from the seizure data that we'll be collecting. He's designed these vectors to be absolutely compatible with what the FDA is looking for human treatment. So at least at that level, that's in place. I think we would really like to see just in the next 3 to 6 months, we'd like to see which of these treatments, GOA versus GOB, maybe a higher dose of the vector will give us better effect than what we saw. Maybe the combined knockdown and overexpression will help. So we want to do a little bit of optimization to try to get better effects in the readout while still avoiding complications like the introducing seizures. So there's a bit of optimization at this level. And then what exactly will be required usually requires a pre-IND discussion with the FDA. Miguel, Dr. Estevez has done that already, so he would know the process around that more than I would. Yeah, so the question is, when I say a lot of money, how much money does it take to get a vector into a patient? Probably on the order of millions. But I think where there are particular, and it may be allele specific. So it may be that this treatment, you validate it for one mutation, and the question came up, how do you validate it for others? It may be that one mutation is present in enough people that you could spread the expense across that group. But I would say definitely on the order of a million, few million. Okay.
Well, thank you, Dr. Newbig. Appreciate your efforts to continue to champion your research and move the ball forward for the GNAO1 community. And thank you for being here today. Really appreciate you. And I'm excited about those red bars too, which is weird for a communicator to say, but the math and the science really excites me. Our next speaker is Brandy Wentland. Brandy is the speech language pathologist. She specializes in augmentative and alternative communication, known as AAC. She provides AAC training for parents and school teams and therapists. And she also conducts AAC evaluations and assists with AAC implementations for social groups and ongoing therapy. While living in Arizona from 2016 to 2021, Brandy hosted an out and about East Valley event where she met Bob Davison, Leanne Atkinson, and their family. So she, and I think Bob and Leanne as well, are going to speak to us today about their experience with AAC, and we'll welcome them to have the conversation.
Thank you, everyone. It's a pleasure to be here today. And I'm really happy to be here with Leanne and her family. As you mentioned, I'm a speech language pathologist and I specialize in augmentative and alternative communication. And I'm joined here with Leanne.
Hi, everybody. I'm Leanne Atkinson, and I Rita gave my, I'm a member of the Davison family pictured here, and I know AAC is just is something very important to us and our family. So I thought of Brandy right away and my experiences with her on the with Out and About, and I think it really, I really wanted her to bring her here for this because of just those experiences. They're very inclusive, and it was children of all ages and all types of devices. It kind of made me think of visiting with you all. So that's what brought me here.
So we also wanted to share with you a little bit about Out and About, and we're going to do that later today. So one of the breakout sessions will be an actual Out and About event so you can bring your children. We're going to have some fun, get you a small slice of an idea of what it was like when we were together in Arizona. But right now, what I'd like to talk to you about is just what is AAC. Some of you may be starting your journey or maybe are further along on your journey. So AAC can be comprised of more things than just a device. Sometimes we think of it as the actual device that you get funded from insurance, but it's more than that. It can be facial expressions. It can be text messages. It can be gestures or sign, right? And so AAC is all these different forms of communication. And honestly, we all use it. We all use it. So we don't need to limit AAC to one mode. We all use multiple modes, so our kids can use multiple modes too. So who needs AAC? This is a picture with my good friends, Krista and Lydia. They both have cerebral palsy and use AAC. They also use all of the modes I talked to you about. They are, Lydia is a college graduate, Krista is still in college, and they use their devices when they're doing presentations or preparing things for college assignments. But when they are talking with me, sometimes they might be texting me, or they might be, Lydia likes to write in the air, and this is how she talks with her family. Krista uses gestures and facial expressions. She also uses some sign language. So they use lots of different forms, and they modify those forms based on who they're talking to. Right. And that's true for Rita as well. How she talks with her family might be different than other people in the community.
Yes, yeah, Rita uses her device. She uses hand gestures and verbalization. So, yep.
Absolutely. So we don't ever want to require someone to only use one mode. We want to allow them to use all of the modes, and they get to pick which one of those things they like to use. My husband's been in construction in the past. I would never tell him that you only get to use your hammer. All right. Or in this situation, you should use your hammer. In this situation, you should use your screwdriver or whatever other tools he may have. He gets to choose which tools in which situation and what's going to be most effective. And the same is true for your children. So for those of you that are here, how many of you have someone in your family who uses AAC? Awesome. And how many of you are finding that this is something that's really effective and has really changed communication? And how many of you are really frustrated with the process? Right. So I want you all to know that you're not alone, right? And that's what's so beautiful about this conference is you get to see one another and talk with one another. And that's what we found with Out and About as well, is we had a place where other families like us, other kids like our children that are communicating this way, get to meet one another, and we get to see that journey and see that it's different for all of us. And sometimes there's a high and sometimes there's a low. Rita used her device a lot in the beginning, and then at some point during Out and About,
Yeah, she didn't want to use it, and we just had to say, that's okay.
yeah. And is she using it again now?
Yes,
Not all the time though, right?
that's right.
And that's okay, right? Like, I just want you all to have that peace, and that's okay. There's going to be ebbs and flows. There's going to be times where they want to and where they don't. And we want them to feel like that's okay too. We don't want to put so much pressure on them that they don't like their device, right? So we want them to know that all forms of communication are okay. There's going to be ebbs and flows. There's going to be times they want to use it and times they don't. In one of those earlier pictures, I showed you my good friend, Krista Howard. She's not my client. Krista and Lydia are my friends. They're not my clients. Krista will tell you that for 5 years, she did not use her device. She had it for 5 years and didn't use it. And when her speech therapist would ask her to use it, she'd throw things at her. Kristen now uses her device and she's an international presenter. She uses it at college. And I have asked her, what was the difference? And she said, a boy. So, she wanted to talk with someone and she finally found the power of communication and why it was important and why all of her other modes weren't enough in one particular situation. And that made the difference. And out and about, we had another young girl close to Rita's age, and she was very shy and didn't want to use her device, but Rita was her motivation, right? Seeing another friend, someone else she could talk to, and someone that talked in a similar way to her really made a difference. Anything else you want to say? So there's lots of different types of AAC out there, and there's aided and unaided. So aided means that you need something else, right? So this could be like high tech or light tech. And I've got some of those over on the table over here and also up in the room where we'll be doing out and about. I had all of this sent here from a lending library so that you guys can touch it. I want you to have a chance to actually touch and feel and see these things. Even if your child already has something, it's nice to see what are the other options, right? You're not going to break it, touch it, play with it, have fun. Don't worry if it speaks, it's okay. So there's all of these aided forms of AAC out there. Then there's unaided, right? You don't need anything other than your own body to use it. Facial expressions, gestures, and sign language. There's so many different types of AAC out there, right? On the far left is a category-based system. So you actually have to know that a dinosaur is an animal, right? You need to know that Pepsi is a drink, and you have to be able to navigate categories to find those different items. And this one on the far left is Touch Chat. It also works on Innova Chat. Rita has used both. The one in the middle here, this is LAMP, I believe, and this is semantic compaction. So anything you do with your foot is going to be under the shoe. So you run with your feet, you jump with your feet, you put on shoes, you tie shoes, you do sports. All of those are going to be under the tennis shoe on this one. The one on the far right, this is a text-based app. So for someone that's already literate, that wants to type to communicate. All of these have word prediction. All of these have the ability to type, but there's just lots and lots of different programs out there. And each program might have a feature or a benefit that's better for your child. There is a wonderful woman named Angela Morad who is retired. She's a speech language pathologist. She's putting together tons and tons and tons of resources out there for free for you to be able to look at what's available and kind of sort through what's there. Symbol-based is this first one up here, and those common ones that are most often used are like Touch Chat, LAMP, Proloquo, Avaz, Cough Drop and Speak for Yourself. These are all programs I've often recommended that are very robust, have plenty of language in them. And I've put links in here. So if you, I believe you will all have access to these slides later, and you should be able to click on any of these links to get more information on each of these. I don't expect any of you to become an expert in all of this. This is what I'm supposed to do. But when you want to know, some of you have had AAC evaluations, right? You've had someone either come to your home or you've gone to a clinic and you're wondering, how did they decide what to try with my kid? Right? What's that myth of why did these ones get tried with her? Well, we do feature matching. We look at all the different features of each of these programs. We look at your child and all of their needs and all of their skills, and we say, which ones are going to come together best? That's in an ideal scenario. Now, I'm a speech language pathologist, but not every speech language pathologist has flown all over the country and taking additional continuing education. So you might have a speech therapist who hasn't done that and just went, I know Proloquo. Let's try that with your kid. That's cool and all, but what if LAMP or Touch Chat would have worked better for them? right? So that's where I wanted you to just know what's out there and know what's available. So if maybe a system isn't working or you don't have enough language for your child, that you know there's more there. And then hopefully you can find someone that has done that research, that has gone and gotten continuing education to better help your child. Not all SLPs specialize in the same areas. So mid-tech is where we're taking some technology. We've got some batteries, but we're not so high tech that we have an app involved. This could involve switches. It could involve the GoTalk that's on the bottom left here, or the Logan Tech that's on the top right here. Mid-tech is nice and all, but it's not robust. So what if you want to say, I hate broccoli? Really, you don't really have that option with a lot of these, right? And so as parents, you're having to guess what your kid wants to say. Well, what if your kid wants to say something that you hadn't thought of? Then there's no tech. No tech is really great. It doesn't have voice output typically. It's paper quite often, and it's often laminated. And in the middle here, you will see this Pixie Pal. This is a product that I've gotten affiliated with that will laminate essentially your paper-based printouts for you. I like to believe that we don't have to just use one system. So we can use this mid-tech, we can use this no-tech, and we can use high-tech all together. This is my good friend, Wiki. He's in Special Olympics. He has a high-tech device, but in Special Olympics, his mom does not want that getting left behind or in between each of the events. So that's where he's using his no-tech, is in those situations, right? So you may have multiple forms of communication for your child at different times, either during the day or different situations. Maybe in a situation where your child has fatigue at the end of the day, you need a different device than you needed in the morning. So lots of reasons I use no tech. Some of the reasons might be that we're in water. So maybe the bathtub, the shower, the pool. I might also use it because I want to model language for kids. If I was going to teach your child Spanish, what language should I speak in? So if you want to teach your kids AAC, what language should you be speaking in? Right? So if you're teaching them that language and helping them develop that language, you should be speaking it too. And how hard is it to be speaking on their device? And how much do they want you touching their device? So if you have a second one that's maybe paper-based, it's a little bit easier to get access to, that you could be modeling while you're speaking. Lots of other reasons that we use light tech. How many of you have been in the sun and you can't see your device? Exactly. So that's a situation where I might also have my light tech with me. PECs. How many of you have tried PECs? Okay, see a few hands up. There is this myth that you have to try PECs first. Not true. Please don't let anyone tell you this. PECs is not robust enough. It lets your child say, I want raisins, but it doesn't let your child say, shut up. I like that. I don't like that. Go away, right? Those are important things. We all need to be able to say that. And why should we have to learn how to say, I want milk, I want cookies, before we say stop? So PECs has its good features, but it's just not enough. And how many of you know a 32-year-old adult that's still using PECs? So why are we starting here? How about we start with robust communication? We start with what your kids need later now. They have access to all the words that they're going to need when they're 32 now, so we can start teaching them where those words are at over time, right? We need a system that's going to grow with them. There are no prerequisites. Please do not ever let someone tell you your child isn't ready for a device. I have used devices with 1-year-olds. I worked in early intervention. I've worked with adults. I've worked the whole spectrum. There are no prerequisites. If your child is alive, they deserve to have a way to communicate. There is research here The prerequisites have been debunked for over 20 years. There's lots of myths out there in the field. I want to equip you and arm you for your IEP meetings. I know how difficult that is for all of you, and I want you to feel like you have someone that has your back. So please let me know if you ever need some research going into those meetings to defend what you know your child deserves. I've had some people tell me that that child doesn't point yet, so they cannot have AAC. Not true. There are so many other ways to access a device. We can use eye gaze. We can use a little microlight switch, which I have on that back table over there. Microlight is so cool. You can even use your cheek just to twitch, and that will activate it. We can put the microlight anywhere. So if your child is able to access anything, think about how do they play? How do they touch things? How do they interact with anything? What's their best mode with anything else they use? And what other ways they're accessing their environment, let's find a way to get them to access their device. You want not only a speech therapist like myself looking at language at your child's AAC evaluation, but also a really awesome OT or PT that knows assistive technology that's going to figure out the access mode and the mount that your child needs. How many of you have tried more than one mount? How many have been frustrated with your mount? So one of the things Leanne and I have talked about is, wouldn't it be awesome if you guys had access to see and touch all this stuff instead of only during that one time when people are showing up for the eval? Wouldn't you like to see that ahead of time? Yeah. So for me, every year I go to Florida to ATIA and I get this huge convention hall and a huge exhibitor hall where I get to touch and see all this stuff. I want that stuff available to you guys too. Did you know every state in the US has a lending library where you can borrow this stuff for free? They'll ship it to you. And they'll even send you a label to ship it back. So I want you guys to have access to this stuff to try it out before you're sitting there trying to make a decision with that team. And by the way, you're part of every team. You should be helping make all of those decisions. So here are some of the different modes of access. Key guards are pretty awesome, especially if you have any kind of movement disorder, because you're able to use that key guard to move your finger along and then insert your finger in to say, that's what I want to say. There are lots of key guards out there. There's not just one type. So upstairs, I have several different key guards up there so I can show you the different kinds. You can kind of get an idea what might work best. In addition to eye gaze, there's also head pointing. I have a good friend, Maddie, that uses a silver dot sticker on his forehead, and he uses that for head pointing. He can input data into an Excel spreadsheet faster than I can and in smaller font than I can even see. So you can get really precise with eye gaze and head pointing. I'm a terrible eye gazer. If your child uses eye gaze, I recommend you try it yourself. It's really, really important. I've tried it. I'm actually not very good at it. The sticker works a little bit better for me. There's obviously switch access. A lot of us are used to the switches you actually have to hit, which is kind of frustrating. Any kind of head array where it's just a proximity switch is a lot easier. There's a lot of great head arrays out there now. We've got one down here. ASL Labs is a really awesome company. I really like their equipment. And then up here on the right is partner assisted scanning. And honestly, even if this isn't your child's primary mode of access, I highly recommend you all learn this, because at the end of the day, when you're fatigued or when you're driving in the car or when the device is in a backpack, this is a really awesome way to do it. This family on the top right here, they have a Facebook page, a YouTube channel. It's called We Speak Pod. This mom has memorized the entire book. While she's driving the car, she can do partner assisted scanning. So it's really, really great. And so basically it's just this sister over here who's going through each word and saying each word one at a time. And then her sister is going to give her some kind of positioning to say, yep, that's it. That's what I wanted to say, right? And so if you memorize this stuff, you can actually do this without a book in your hand, and your child will learn that motor plan, and will learn that you're eventually going to get to what it is they want to say. Anything you want to add so far? Okay. All right. So with direct selection, that is where you are directly accessing the device, and that includes eye gaze. With indirect selection, there's something between you and the device, maybe like a switch. And here are a couple of links for lending libraries. They both give you the same information, just in a different format. So I wanted you to have links to both options, but every single state is listed here and what they have available. The Missouri Lending Library lent us quite a bit of equipment today for you to all try out. So I did put some information about them up here, and I have flyers upstairs when you come to the breakout room if you want more information for that. So again, this is Lydia Krista saying, there's not just one mode. Please respect every way that I communicate. Please do not allow any therapist, teacher, anybody to say that your child only needs to use one mode. If I try to treat all of the clients that I work with the same way I do Krista. If Krista says something and I didn't understand, I just say that. Sorry, I didn't understand. Could you find another way to tell me? Right? And so again, giving respect to the individual as to which way they're going to clarify their communication. The most important thing is that your child gets to say what they want, when they want, and however they want. We don't want them to say what's in our head. We want them to say what's in their head. I get so frustrated when I see therapists who do activities like, what color is my shirt? What's this? What's this? Lots of questioning, labeling. Who wants to do that? I don't. That's not how I talk to my friends, right? So therapy should be more of a conversation. It should be a hangout session. It should be a chance where your child gets to talk about whatever it is they want, where the things that motivate them is what we're talking about. So at Out and About, we model that. And at this situation, it was our first Out and About. We were at a pizza parlor, and I purposely picked one that has crazy toppings on pizza, like meatballs and macaroni and cheese, because I wanted these kids to be able to say what they wanted as a topping and it to be something unpredictable, so that these parents could see, like, what would your child say in this situation? Well, this little girl said, I want donut pizza. Would anyone have thought to put that phrase on a device? If she had only a phrase-based system, could she have said that? So we need to have systems that have individual words, and your child knows where all those words are at so that they can make a sentence like this. And that's what we call SNUG, right? It was spontaneous novel utterance generation. So what do you think mom did after she left the pizza parlor? That's right, the donut shop, right? Take your leftover pizza, stop by the donut shop. We're going to go home and eat something really gross. We're going to put donuts on our pizza. And now your child knows the power of communication and knows that what I said matters to you. You heard me, and we're going to go do that. So where do we start? Where do I start? Almost all of my therapy goals, at least the first goal, is refusal. These kids are controlled all the time. They have other people doing things with their body, telling them what to do, requiring them to answer a bunch of yes, no questions. They want to say, stop it, go away, I don't like it. And we need to empower them to say that, especially if other people are controlling their body. So I personally feel that that should always be a number one goal for communication. The next thing I do is I look at what motivates you. What's fun? Whatever it is, we're going to get into that, and we're going to scaffold from there. I'm going to figure out what you want to say, help you say that, and then we're going to start expanding communication from there. So when I was a kid, I went around saying supercalifragilisticexpialidocious a lot. And I think anyone should have the right to say that, but that's probably not what kids say nowadays. So what did you guys program on Rita's device?
So on the first one she had, we did program Shut Up for her to use. She didn't really use that, but I wanted her to have that option to say it. And just most recently, we're originally from Wisconsin, I am, and I added "ope" to her current device. I don't know if she'll use it, but yeah, I want her to be able to say what she wants to say.
So my mentor, Dr. Caroline Musselwhite, she's a leader in the field of AAC. If you don't know who she is and your child's using AAC, I highly recommend looking into her, but she calls them juicy words. right? So get a communication circle with some other kids, some other peers, and ask them, what are some words you say that aren't probably on here? What are some words that aren't in the dictionary? Maybe they're in the urban dictionary, right? What should we put on here? So, I shouldn't be in charge of that because I'd probably put something like a lit and kids would tell me that's not a thing anymore. So, you need to have their peers tell you what to put on the device. So I know a lot of you have had AAC evaluations, or some of you have some upcoming ones. You have the right to have one once every 3 years. Your child can be reevaluated for a new one. If your device isn't working for you, that's happened before, right?
Yes, so I guess our experience was, so that on that picture is, we just touch chat. It's on an iPad Air. That iPad Air just couldn't take the updates anymore. And now we have a new device. We went through the eval, got a new device. So, I didn't think of that when we got the device, like we'll be doing this over and over again.
Absolutely. So I just want to equip you of what that looks like and get you kind of an inside look of what we're looking at as evaluators. I did evaluations all over the state of Arizona, and believe it or not, I've also done evaluations in other states, and they're different from state to state. So what's true in Massachusetts is very different to what's true in, say, California. So what is common is that there's different funding sources. Your insurance is one of those funding sources. Your school district, according to FAPE, should be requiring you one as well. And technically, you could get one from each. Now, if you get one from each, you sure hope that those evaluators agree on the same device. Because I've had families who had one device at home through insurance, and the school recommended another one and refused to implement the one from home. That's frustrating, right? And then there's private pay. For a family whose child is using direct select with their finger and doesn't need a key guard or any accessories or any mounts, that works. But if you're having to get an eye gaze device or a mount or something, that can get pretty pricey. So I generally recommend going through insurance or school district, but there are ways for you to get some of these apps on an iPad you already have at home, at least as a backup. So that is an option. Then when they come to do the evaluation, either in your home or you go to a clinic to do this, or maybe it's done at the school, they should be trialing at least 3 devices. And you could talk to them about that ahead of time. Not a lot of people will tell you that, but it's a great idea because you could explore some of these systems before they show up. Because I don't know about you, do you kind of freeze up during those situations? It's a lot of information all at once. So anything you can get ahead of time helps prepare you. So you could talk to them about, what are some of the devices you're thinking of trialing with my child? Then there's the feature matching. Like I said, where we're looking at, how is your child accessing the device? What is the type of device? What type of system, like software, are we going to be using? How many buttons per page? And there could be a trial period. In Arizona, generally, there's no trial period. They come, they see you one time, they recommend it. 1 to 6 months later, you get a device that shows up, and hope that everybody picked the right thing, right? In other states, you get to try it for 30 days and decide if you're going to go with that or not. So there's lots of different options out there, and I just want you to know kind of what your rights are and what the range is, so that you're a little more prepared the next time you have an AAC evaluation. There are lots of different companies out there. These 3 companies tend to have reps in your area that will come to you or will come to the school, that will provide free training. These companies have boots on the ground. So I just threw some links up there so that you can click those links and then find your local rep and talk to them. Some reps are amazeballs. I love them. They've done a lot of training with me and school teams. Other reps, not so great. So just want you to know what's out there and contact them all. Talk to all the reps. Have them bring stuff out. Have them try it with you. Have them come train your school team.
Thanks. I don't know. I mean, we've just had so many experiences with AAC and the device, and I guess, like the most recent eval, I knew I wanted Rita to try eye gaze, and and it's probably from coming to the last conference, I was like, I think we need to try it. And it didn't work. But at least it was on the list. Cause I think initially they were like, oh, I don't know if that's right. And I was like, no, I want to try it. And we did. And we're sticking with what we got the Nova chat. So that's what
Yeah, yeah. And I want to empower you guys to ask those questions, right? Like, I think a lot of times you sit at these IEP meetings or in medical appointments and you see all these professionals sitting around you, but you're the professional in your child. You have a PhD in your child and you know so much more than you give yourself credit for. And I think a lot of times we go into this default of, the professionals know, but sometimes you know better than us. There are those times where you do, where you know something we hadn't thought of, where you know something because we don't see your child 24/7. Right. And so I just want to empower you to have your voice that that's in there, that voice that wants to get out during a meeting and wants to ask a question or wants to challenge a professional. Go ahead and ask that. If you want to see eye gaze, even if the professionals are saying, I don't think it's going to work, say, you know what, just rule it out for me, just so I know. Right. You have that right. And I want to empower you to have those rights. My contact information is up here. I am licensed in a few states. I can't do work in states I'm not licensed in, but I can still help you. I can help you find people. So if you're looking for a really good AAC evaluator, or you're looking for a speech therapist that knows AAC, feel free to reach out to me. I know people all over the nation. I'm more than happy to help you find someone that's good, that's done continuing education, and answer any questions that you may have going into any of these evaluations or IEP meetings. Do you have any questions? Yep. Yeah. Oh, absolutely. The question is, how do you know when a device is outdated? And I would say typically insurance companies allow you to get a reevaluation every 3 years. So I would take advantage of that every 3 years. You just have to give them medical justification for the reevaluation. And so you need to say kind of what you just said. I work in the tech industry and devices often are not working. They're not updating. They're glitching. And we had to do that with Rita's device as well, where we had to explain why, no, it's not broken, but we're not going to wait until it's broken to get a reevaluation. We want to get this now, and you don't have to return the old one. So what's nice is the old one can become your backup when you get the new one. Really good question. Yeah. Yeah. Is it easier to get a subsequent AAC evaluation approved than the first one? No. They all can be difficult. A lot of times it's how the report is written and how good that speech therapist is at writing a revision to get that approved, and how well they know the insurance companies in their state. I usually recommend going through some of those companies that I listed their links, because they have huge funding departments and they know what works in your state, and they will help with that rewriting process and saying, hey, this section right here is probably why it's getting denied. Can you help rewrite that and resubmit that? And that's when we're dealing with insurance. The state is a whole other ballgame. If you're trying to get state approval, Arizona, we had our really interesting scenario a year back. Every funding source is different, and school districts can be really challenging. And that's really just a school district to school district situation on how they read FAPE and what they believe FAPE should be providing. Absolutely. I'm available all day. Stop me, grab me whenever. And I'm definitely available during the breakout session. It's going to be very interactive. I want you to bring your kids. I want to have fun. I will have a few slides to kind of tell you what Out and About is, but for the most part, I want it to be a place where it's casual and a good place for us to all have a good time, the kids to have fun, but also for you to ask questions. And anytime you see me, just grab me. I'm more than happy to talk. Yes. Really good question. So the question was, when do you progress to a text-based system? And I would say generally you don't. Generally, you start with a system and stay with that system. It's kind of like we don't generally progress from English to Spanish, right? You think of it more like a language system that they're going to learn. The only time we change a system is if a system's not working for someone or someone identifies they need something else. Typically, all of these picture-based systems have a keyboard. So you can already use that keyboard. And actually, it's faster to use picture-based than it is to use text-based. So if you think of a word like should, how many clicks to spell that word should, versus if you're using picture-based and you know right where it's at, it's probably only 2 or 3 hits, especially if you're using like LAMP Words for Life. Nothing is more than a 3-button hit. It was created by somebody that's a court reporter and a linguist. So they took both of their skill sets and said, let's make something that's as fast as possible for someone to communicate. Really great questions. So the question is, what happens when we start getting into the upper grades and she's having to write essays, right? As I mentioned, 2 of my friends, they've been through college. So their systems, they both, Lydia uses LAMP, like your child, Krista uses Unity, which is both from PRC. And those systems can be plugged into a computer and you can export anything that they type on their system. Also, you want to get the assistive technology team at the school involved, because in addition to their communication system, they should have a bunch of other stuff too on the computer, right? And so like Krista does some of her stuff on her Unity device, and other things she uses like SnapType and other apps from the assistive technology team to help her figure out what to do to write her papers. She really likes to have like a thesaurus and other things like that as well. So it goes back to that multimodal, but you can use the picture base to create a full paragraph. Krista composes all of her speeches when she presents full hour presentations on her device and stores them to be able to communicate. Other question? Krista likes Co-Writer a lot. She does that on her computer. I don't believe there's a way to use Co-Writer and the AAC app at the same time, but she uses Co-Writer with like Word or something like that on her computer. Absolutely. So in the back, she asked, could they use Co-Writer? And she's an occupational therapist. Love OTs. My mentor is also an OT. So Co-Writer is a program that uses assistive technology to help create any kind of word processing. And it has a lot of additional features in there to help you with that. For Krista, it really helps with her spell check and thesaurus and some other features. Anything else you want me to highlight with that? Yeah. Can I ask you to repeat it one more time with a mic?
Yep. So what I use it for a lot in occupational therapy is to be able to use it for kids who really want to write about a topic. So the way it works is it's a word finding thing. So if your child was writing a story about dinosaurs, you would have the child start with the couple words that they know, like I am, for example, in the jungle, I saw and you want to say brontosaurus, you would write the first letter B and it would bring up every dinosaur that has the name started with B. And it would continue on, then you could type another word or two that he knows or she knows, and then he would progress it to being able to then find the next word that they may be struggling to actually type. So it just helps them be able to pull up words. And if you look it up, you can see it has actually video tutorial on the system. It is a costly system. That's the only negative. So it does cost I don't know off the top of my head how much.
But
it
the
is.
school should pay for that.
And the school does pay for it. I have found many schools to have covered this.
Yeah, they really should be paying for that. And if your child is transitioning, then vocational rehabilitation should be paying for that. The conference I mentioned earlier too, ATIA, it's open to parents and professionals. It's in Florida every year in Orlando, and it's on assistive technology and AAC. So all of you that are here would benefit from seminars that are there and also from seeing all of this stuff in person at the vendor hall. You can come to the multi-day conference, or you can come to the one day just to come to the vendor hall and check out some of the stuff. It's probably my favorite conference. There's another one similar to it called Closing the Gap in Minneapolis, Minnesota, and I believe that one is in October. Any other questions? Thank you.
Well, thank you, Brandy. Thank you, Leanne. Appreciate y'all being here. I know the breakout session will be heavily subscribed this afternoon, I'm sure.